Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Assalamu alaikum, dear class fellows, friends, and respected teacher. My name is Kifayatullah. I am the student of BA 7th Microbiology from Kohat University of Science and Technology. Welcome to my presentation. This is the front page of research paper from where I prepared my presentation topic, and my presentation topic is molecular study of the prevalence of Helicobacter pylori infection and its resistance to antibiotics in Constantine, Algeria. Contents. These are the contents of my presentation topic: introduction, objectives, materials and methods, results, discussion, summary, and references. Introduction. Helicobacter pylori, first described by two scientists named Warren and Marshall from Australia, and it is gram-negative spiral-shaped bacterium. It is important human pathogen that colonized the gastric mucosa, and estimated about 50% of world population is colonized. So it causes gastroduodenal diseases such as gastritis, peptic ulcer, adenocarcinoma, and gastric malt lymphoma. Triple therapy named standard therapy is used as first line of treatment for H. pylori worldwide. So triple therapy include a proton pump inhibitor, amoxicillin, and clarithromycin. However, this treatment may fail due to several reasons. The most important one is H. pylori resistance to clarithromycin. Objectives. The objectives of this study were to evaluate the prevalence of H. pylori infection, to detect the mutation associated with resistance to clarithromycin and fluoroquinolones by using molecular method for the first time in Constantine, Eastern Algeria. Material and methods. Collection of gastric biopsy samples. Gastric biopsy samples were obtained from antrum of adult patients and submitted to upper gastrointestinal endoscopy at the University Hospital of Constantine in Eastern Algeria. One piece of each sample was stored at -80 degrees centigrade and sent to the National Reference Center for Campylobacter and Helicobacter and Bordeaux, France for molecular study. Molecular methods. So we have two molecular methods: DNA extraction, and the second one is real-time PCR DNA extraction. Each gastric biopsy sample was digested in 180 microliter of lysis buffer and 20 microliter of proteinase K and incubated at 56 degrees centigrade for 5 hours. Genomic DNA was isolated using a MAGNA Pure LC DNA isolation kit 1. Real-time PCR. Detection of H. pylori infection and determination of point mutation associated with clarithromycin resistance in the 23SR RNA gene performed by real-time PCR. This method is based on the amplification of fragment of the 23SR RNA gene of H. pylori, and it followed by simultaneous detection of the product by probe hybridization using fluorescence resonance energy transfer technology. The quinolone resistant determining region of the H. pylori garage agent was detected by standard PCR. The PCR product sequence on both strands using the same primers. Results. 135 Algerian patients aged 16 to 84 years were included. There were 69 females, that is 51.1% females, and 66 males, that is 48.8% males. 33 patients with normal mucosa, 90 patients with chronic gastric, 4 with gastric ulcer, 3 with gastric adenocarcinoma, and 5 that is 3.7% with duodenitis. Detection of Helicobacter pylori infection and determination of clarithromycin resistance. H. pylori DNA was detected in 86 gastric biopsies from 135 patients, 44 that is 51.1% from males and 42 that is 48.8% from males. So the below table 1 showed that point mutation conferring resistance to clarithromycin and Helicobacter pylori according to result of 23 srRNA mutation. Detection of point mutation associated with quinolone resistance. PCR amplification and sequence analysis of the quinolone resistance determining region of gyrase A gene from 84 DNA samples revealed that the resistance rate of H. pylori to quinolone was 6%. So the below table 2 show that among the 5 resistant strain, 1 carried double mutation while rest of the 4 had single mutation. Discussion. In Algeria, the clarithromycin-based empirical therapy is widely used as first line of treatment of H. pylori infection. The present study is the first to report a primary resistance rate to clarithromycin reaching 18.6% in Constantine, eastern Algeria. This rate of resistance to clarithromycin was related especially to the genotype A2142/43G. Although fluoroquinolones are not used in the treatment to eradicate H. pylori infection in Algeria. Summary. The main interest of this study is to provide important data on the status of H. pylori prevalence and local antibiotic resistance rates in Constantine, eastern Algeria. This study reported an increasing rate of primary resistance to clarithromycin, that is 18.6%. So the whole summary means that the standard therapy clarithromycin based triple therapy is used as a first line of treatment for H. pylori infection. These are the references of my presentation topic. Thank you.